So as far as NEET is concerned, you'll get 3 questions from this chapter, Kingdom Animalia. So anyway, we are late. I'm going straight away into the class. Now, Kingdom Animalia or Animal Kingdom. I think you might study the classification. How many kingdoms are found according to Linnaeus? Or you tell me how many kingdoms are found or how many kingdoms are there according to Whitaker classification?
5, sir.
5 kingdoms.
5 kingdoms.
Yes,
What are
sir.
those 5 kingdoms?
Kingdom Monera, Kingdom
Very good.
Protesta,
Monera,
Kingdom Fungi, Kingdom Plantae,
Protista,
Kingdom Animalia.
fungi,
Plantae and
plantae,
animal.
and animalia. So these are the 5 kingdoms according to Whitaker. Now, what are the living organisms included in Monera?
Bacteria.
They are simply called as prokaryotes. Best example, bacteria. And we can call them as unicellular decomposers. Then you tell me, Protista.
Unicellular eukaryotic organisms.
Very good. They are unicellular eukaryotic organisms. Can you tell me some examples?
Amoeba, paramecium.
Very good. Amoeba, paramecium, euglena, all these organisms come under Protista. Then you tell me what are the organisms included in kingdom fungi? Multicellular decomposers. Then what are the organisms included in plantae? Multicellular autotrophs or producers. That means all plants were included in kingdom plantae. And what are the animals included in kingdom animalia? All multicellular consumers, are what we call them as autotrophs, and these are heterotrophs. Got it? Now, today we are going to discuss about the kingdom Animalia. Now, I'll tell you another name of kingdom Animalia. It is metazoa. Just note down. The other name of kingdom Animalia, metazoa. When you look at this kingdom Animalia or metazoa, we can find different animals who belong to different phyla. I think you might heard about the phylums: Porifera, Cnidaria, Ctenophora, Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. So total in your textbook 11 phylums are given. I'll repeat once again. Phylum Porifera, phylum Nidiria, Ctenophora, Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. Total 11 phylums. Among the 11 phylums, these 10 phyla are considered as non-chordates. Why? Because they are not having backbone called notochord. And these are chordates who are having backbone. So different animals were included in different groups. So just look at here once. Total, how many non-chordate phyla are present? 10. Along with these 10, we can observe many minor phyla, but all these phyla are not required at this moment. We are talking about the major phyla. So we are having 11 phyla, among them 10 non-chordates and 1 chordate. Just look at here. When you look at the living organisms all over the globe, we can find millions, trillions of animals, isn't it? I'm not talking about the number, I'm talking about the species. Millions of species are present all over the world. Even though we are having millions of organisms, even though they are showing great diversity, what is meant by diversity? Differences. Even though they are showing great diversity, all these organisms were placed under these 11 phyla. Just look at the wonderful thing which is did by, and all this work is done by taxonomists. And how we can keep all these millions of organisms on only 11 phyla? Based on some similarities. Even though they show great diversity, they show some fundamental similarities, like levels of organization, symmetry, coelom, and pattern of different systems like digestion, respiration, circulation, etc. See the wonder. Even though we have millions or billions of organisms, and all these organisms, they show some fundamental similarities like levels of organisms, symmetry, coelom. Now let us see this one by one. First, levels of organism. Levels of organization. Now here, based on the complexity of the body, we can divide this levels into cellular level of organization, tissue level of organization, organ level of organization, and organ system level of organization. Cellular level, tissue level, organ, and organ system level. Now, what is this cellular level? No doubt, these animals are multicellular. They are having different type of cells who performing different functions. But even though the cells are performing different functions, there is no coordination between the cells. I'll repeat once again. The cellular level of organization is found in Porifera, phylum Porifera. Example, sponges. What is the type of organization found in sponges cellular level? Very, very important. They are the first metazoans, that means first multicellular animals. First multicellular animals made up of many cells. And we can find different types of cells who perform different functions. There in NCERT, one line was given. They show division of labor. Division of labor. What is meant by division of labor? Different works were shared by different cells. And we can find different type of cells. Some perform the process of digestion. I'll give a brief explanation. This is a sponge. We can find different type of flagellated cells. And I think you might study it is having small openings all over the body called ostia. The inlet openings are called ostia. And continuously the water move into the body through ostia. And the water current is set by the flagellary beating of the flagellated cells. What are those flagellated cells? What is the name of the particular cell? We'll discuss later. Now, one of the type of cell, flagellated cell, help in creating the water currents. Number two, it help in the process of digestion. Likewise, we have different other cells like archaeocytes, thesiocytes, pinacocytes. Like that, we have different type of cells who perform different functions. That means different activities are carried out by different cells. So here we can find division of labor. Is it okay? Now, even though these flagellated cells are performing the same function, there is no coordination between any two adjacent cells. That means these cells, they work individually, but not collectively. Their work is not coordinated because of the absence of very important, because of the absence of nerve cells. Why there is no coordination between the cells of Porifera? Absence of nerve cells. And this type of arrangement is called loose cell aggregation. Aggregation. What is meant by aggregation? Together. Cells are loosely arranged. What is meant by loosely arranged here? There is no coordination. They are not working together. They are working individually because of the absence of nerve cells. So even though it is multicellular, there is no coordination. So the cells are doing their job individually. So the name given as cellular level. The job is done at cell level. Next example, Porifera. Number two, do you have textbooks with you? Do you have NCERT textbooks with you?
Yes, sir.
Yeah, you
Yes,
can
sir.
open the textbook and you can see the textbook. I'm just explaining almost from textbook, and that will be useful to you in next week examination. And after the completion of this unit in next week, we can discuss the NEET level. Right, Amma. Next, coming to tissue level. The tissue level of organization is found in Coelenterata. Other name of Coelenterata, Nidaria. Now, what happened in Coelenterata? Some of the cells who performing a particular function started doing the work collectively. So here we can find coordination between the cells. And this coordination is brought by the development of nerve cells, very important. Due to the development of nerve cells, very, very, very important. So here, nerve cells, note down, nerve cells first appeared in Nidaria or Coelenterata. Now, a group of cells, very, very important, the group of cells performing a particular function together developed into tissue. So tissues first appeared in Coelenterata. And one thing, remember students, you have to follow my teaching carefully. In the middle of the teaching, I'll ask questions individual. That's why I ask you to on the videos. I can see each and every student and I can ask questions. Are you ready? Simply sitting and listening the class will not give any result. And you must understand the concept. If you have concept, automatically you'll get success. Is it clear? Right, Amma. So let me complete this. After the completion of this part, I'll ask questions. Get ready. Tissues first appeared in Coelenterata or Nidaria. The cells performing a particular functions together called as tissue. And a group of tissues together performing a function is called organ. Best example, heart, kidney, liver, like that. I think you know. Heart is made up of different tissues like epithelium, muscular, nervous tissue, etc. A group of tissues together performing a particular function is called organ. Now here, organ level of organization first appeared in Platyhelminthes. Platyhelminthes. And a group of organs together performing a particular function is organ system. For example, circulatory system. Amma, what are the components of circulatory system? Heart, blood vessels, blood. Different organs, they are working together in a coordinated fashion to perform a function is called organ system. Best example, digestive system, circulatory system, excretory system, nerve system, like that. Now, organ system level of organization first appeared in, very, very important, Platyhelminthes. But well developed from Aschelminthes. But in textbook, it is given the organ system level of organization is seen or well developed in Annelida, Arthropoda, Mollusca, Echinodermata, Chordata. But remember, very important, organ level of organization first appeared in Platyhelminthes. Organ system level of organization first appeared in Platy, but well developed right from Ascalmentis. Right. Now, I'll ask few questions. Levin,
Yes, sir.
dash level of organization is most primitive type of organization.
Sir, I couldn't hear you. Can you repeat it, sir?
Which level of organization is most simple type of organization or primitive organization?
Cellular.
Very good. And what might be the reason for the loose aggregation of cells?
Because there's no nerve cell.
Very good, very good. And in which group of animals first time tissues develop?
Coelenterata.
Very good. What is the other name of coelenterata?
It's that Cnidaria or something, sir.
Cnidaria
I'm
or
not able to pronounce
coelenterata.
it.
Very good. In which group of animals organ level of organization first appeared?
Platyhelminthes.
Very good. And organ system level of organization is well developed from?
Ascalmentis.
Very good. Very good. Next. So, what is the more complex organization here?
Organ system.
Organ system. Very good. Next here, in this organ system level of organization, we can find more complexity. Even though they show some similarities with respect to digestive system, respiratory system, you just look at here. When you look at the respiratory system, right from fishes to mammals, or circulatory system, right from fishes to mammal, you can see some similarity. At the same time, small differences we can find. Now let us see the digestive system of animals, right from platy to chordata. In some animals, we can observe the development of gut. What is meant by gut here? Gut. Gut means alimentary canal. Now, in Platyhelminthes, gut is sac-like, very important, or incomplete. Gut is sac-like or incomplete. So that means mouth is developed, but the alimentary canal is not complete, not having the opposite opening, that is anus. So here we can find a single opening, and the same opening acts as mouth as well as anus. So through this opening, it can take food, and after the digestion, the waste material can be sent out. So sac-like or incomplete gut is found in platy. And what are the next phylum after Platyhelminthes? Ascalmentis. Right from the Ascalmentis, we can observe the development of complete gut, starting with the mouth and ends with the opening, anus. This is mouth, and this is anus. Okay, ma? So here, complete get. If you have any doubts, you can ask me freely, okay? So it starts with mouth, ends with anus. And just look at here. This level of organization is also called as tube within tube organization. And you can see this tube. How you can say this tube within tube organization? Just imagine body is like tube. Within elementary canal is another tube. This is called tube within tube organization. And here tube within tube organization is seen right from ascalementhes to chordata. This is one small difference we can observe among the animals with respect to digestive system. And coming to circulatory system, here we can find two types of circulatory patterns. Number one, open type. Number two, closed type. Open type of circulation and closed type of circulation. Open type and closed type. What is the difference between open type and closed type? We'll see. Open type. In these animals, no doubt we can observe heart. Here also we can find heart. And I think you know what is the function of heart, Amma? To pump the blood, isn't it? Now this heart opens into, or it pumps the blood through main big vessel called artery. Here also the blood is pumped to the body parts through artery. So the blood generally reaches the tissues through artery. And in case of open type, the blood from the artery directly move into tissues or body cavity. So here these are all organs, and this is blood. This is blood. The blood that comes through arteries directly released into body cavity. And here one word is given in NCERT, cells and tissues are bathed in blood. That means they are in direct contact with the blood. And the blood that receives the waste material can be collected through a vein and again sent back to heart. Like that, you can find the circulation of blood. And this is called body cavity, cavities. Whereas in case of closed type of circulation, the blood that comes out through arteries generally passes through still small vessels called capillaries. And all these capillaries again join together form vein. See, the blood never come out from the blood vessels. Then how tissues and cells get nutrients and gases? See, I think you know capillaries are very thin, made up of a single layer. While the blood is passing through, see the wonder here. When the blood is passing through capillaries, you can observe diffusion of nutrients, water, gases to the surrounding tissue. And the waste materials released by the cells can be taken up by the blood and returned back to heart through veins. So in case of closed type of circulation, the blood never come out from the vessels. It passes through the vessels of varying diameter. For example, artery is large, while it is passing down towards the tissues, the diameter gradually decreases. And again, these small vessels, they combine and join together, form veins. Still, the diameter is increasing and carry the blood towards heart. Okay, ma? So this is the difference between open type and closed type. According to NCRT, open type of circulation is given for arthropoda, mollusca, hemichordata. Arthropoda, mollusca, even echinoderm also having open type that we'll discuss later. Closed type. Annelida, chordata. Annelida chordata. And here we can add some more phyla while we're discussing the different phyla. So this is about open type, closed type. So this is about levels of organization. Did you understand this? Any doubts, Sama? Right. Evelini, what is your name?
Evelyn, sir.
Amma?
Evelyn.
Evelyn.
Yes, sir.
Evelyn. Tell me. What is the main difference between open type and closed type of circulation?
Sir, in open type, the cells are in direct contact with the blood which flows from the arteries.
Very
and in
good.
closed type, the blood only flows through the capillaries. The blood doesn't come out from the blood vessels.
Very good. Very good. In which group of animals we can find closed type of circulation?
Closed type is in chordata and annelida.
Very good. Good. Nice. Next, Amma, coming to coelom. Coelom. What is meant by coelom? Body cavity. If we take an organism, see, this is an animal, mouth, anus. I'm just taking cross section of this animal. This is cross section. When you take cross section, you can see that animal like this. What is the outer circle? The body wall. Inner circle, elementary canal. Elementary canal. Between body wall and elementary canal, we can find different organs. The organs which are found within the body cavity are called visceral organs. All visceral organs. Now I think just keep this coelom aside for 1 minute Let us see the germ layers found in living organisms. What are germ layers? You tell me, anyone? Germ layers. Actually, during embryonic development, I think you know, our life starts with a single cell called zygote. And that zygote undergo cleavages, means divisions, and form a ball called morula. Then you can find development of another stage called blastocyst. Blastocyst. Are you first year students or second year students, +1 students or +2 students?
First PUC, sir.
First PUC. Okay. So zygote develop into morula and then blastocyst. In this stage, we can find a mass of cells. This is called inner cell mass, and this cell mass differentiate into 3 layers during growth as ectoderm, endoderm, and mesoderm. I think you got it. I'll repeat once again. During embryonic development, a single zygote multiplies and form a mass of cells. I'm just explaining in a simple manner because you don't know the actual developmental pattern. Now, this is a mass of cells. And here the mass of cells all look alike in the initial stages. But later on, they undergo specialization and differentiate, or they transform into 3 different types of cells. They are called ectoderm, endoderm, and mesoderm. In some animals, we can find only 2 germ layers, like ectoderm and endoderm. Between these 2, we can find a jelly-like, very, very important jelly-like material called mesoglia. Glue means that attaches the 2 layers. It's a glue-like substance that can attach the 2 layers. Those who are having 2 germ layers, germ layer is one which can give rise to different organs. So our entire body was derived from only from 3 germ layers. Some organs are derived from ectoderm. Best example, skin, nerve system. All these are derived from ectoderm. Some organs are derived from mesoderm, like muscles, bone, blood, etc. Some structures are derived from endoderm, like inner part of alimentary canal. So likewise, our entire body is derived from only 3 layers, and what we call these layers as germ layers. How many germ layers are found in lower group of animals like Cnidaria and another group Ctenophora, 2 germ layers. So they are called diploblastic animals. I'm just explaining the para below coelom. Without understanding these germ layers, you can't understand the coelom. I'm just explaining the next para to coelom. Diploblastica, triploblastica is given. Those who are having 3 germ layers, like ectoderm, endoderm, mesoderm, ecto, endo, mesoderm, are called triploblastic animals. They are called triploblastic animals. Who are diploblastic animals? Cnidaria and Ctenophora. Triploblastic animals, Platyhelminthes to chordata. Platyhelminthes to chordata.
Excuse me, sir.
Ma.
Sir, are sponges also diploblastic?
No. Where the cells are not arranged into germ layers. Even though they are having 2 layers, we can find 2 layers, pinacoderm, coinoderm. But even though the cells are arranged in 2 layers, we should not call them as diploblastic. Why? Because there is no coordination between the cells. They are still at a cellular level. So in which group of animals we can find a diploblastic condition? Cnidaria and Ctenophora. Triploblastic condition, Platyhelminthes to chordata. Okay, na? Platyhelminthes to chordata. Now let us go to coelom. I told you coelom is a cavity found between body wall and alimentary canal. The outer layer is called ectoderm. Inner layer is called endoderm. Actually, how this mouth is formed, how anus is formed, based on the development of mouth, animals were classified into protostomia, deuterostomia, and we can find different types of coeloms. Everything I'll explain after the completion of examination. Okay? If I explain everything, you will get confused. Right, Amma? See, this is ectoderm and this is endoderm. Between, we can find visceral organs. Viscera means internal organ. See, look at here. Ectoderm develop, endoderm develop, mesoderm. Now here, in some animals, I'm showing 3 different group of animals. 1, 2, 3. Gut. This is platyhelminth. This is platy. This is aschelminth. And this is from Annelida to chordata. Annelida to chordata. Come on, look at here. First, let us see platy. In platyhelminthes, the mesoderm that is developed fills the total cavity. So it does not leave any space or cavity. Now the mesodermal tissue completely fills the cavity found between ectoderm, endoderm, and visual organs. And this tissue is called mesenchyme or parenchyme. Mesenchyme or parenchyme. So these animals are not having any body cavity. Total cavity is filled with tissue. So they are called acoelomates. They are called acoelomates.
So can you explain this again?
Yes, I'll
sir?
explain once again. In case of platyhelminths, the mesoderm that is derived, mesodermal tissue that is derived from mesoderm fills the total cavity. Fills the total cavity, leaving behind no space. As the space is absent or cavity is absent, the name given as acoelomate. No coelom, no cavity. Number 2. In case of Aschelminthis, see, ectoderm, endoderm, but mesoderm is scattered means spreaded in the form of pouches called mesodermal pouches. Here there is a cavity between body wall and alimentary canal, but that cavity has no special lining, so they are called pseudocoelomates. Pseudocoelomates. In some animals, no doubt there is a cavity, and very, very well, look at here, this cavity is lined by mesodermal layer. What is that mesodermal layer? I'll tell you later in need. See, this layer surrounds the body wall, alimentary canal, as well as visual organs. This is mesodermal layer. Mesodermal layers, also called as peritoneum. Also called as peritoneum. Okay, ma? So, according to NCERT, I'll tell you. The animals with the space found between body wall, alimentary canal, lined by mesodermal layer is called coelom, or it is also called as eucoelom. What is meant by eucoelom? True coelom. True coelomates. The first true coelomates annelids. Number 2. In some animals, there is a space, but the body wall, alimentary canal are not lined by mesodermal layer. But the mesoderm is scattered in the form of small pouches. In platyhelminths, total cavity is filled with tissue. And that tissue is called mesenchyme. So they are acoelomates. I think you understand the difference between acoelom, pseudocoelom, and eucoelom. Do you have any doubts? Amma? Are you following my teaching properly? Is it okay for you? You want me to change any teaching pattern? Is it okay? Are you comfortable with my teaching? Sure. Very good, very good. So we completed the C law. Next, coming to symmetry. Now, what is symmetry? Division of body into two equal halves through a particular plane passing through the axis is called symmetry. Amma, what is the difference between axis and a plane? Can anybody tell me the difference between axis and plane? Axis is an imaginary straight line. Note down. Axis is an imaginary straight line that extends between two ends or two surfaces.
Sir, can you please repeat?
Yeah. Is an imaginary straight line that extend between two ends or surfaces. What is this imaginary line? I'll tell you. When your mother wants to, suppose your mother is given one apple to cut into two equal halves, then what you will do? You first, you will imagine a line. You will imagine a straight line. Then you will try to cut that apple in order to get two equal halves. If this cutting passes through that imaginary line, then only you'll get two equal halves. Otherwise, you can't get the two equal halves. Just let me give one more example. Just imagine myself is an organism. Suppose if you want to cut myself into two equal halves, how it is possible? First of all, we'll imagine a line that extends between anterior to posterior, central line, what we call it as the principal axis. Principal axis. If you cut the animal through a particular plane, you just note down. Plane is any flat surface that runs through an axis. That runs through an axis. A flat surface that runs through an axis is called plane. Now I'll tell you one example. This is mouse. I want to cut this mouse into two equal halves. And first of all, I'll imagine a line that extends between this end to this end, what we call it as a principal axis. Then I'll try to cut this mouse into two equal halves. The flat surface or any surface that runs through an axis is called plane. Now you tell me one thing. Through how many planes I can cut this mouse into two equal halves? You tell me. I think you understand the difference between axis and plane. I'll tell you with simple example. Take one water bottle, round steel water bottle. And this steel water bottle will have upper and lower ends, but no ventral, dorsal, lateral surfaces. We have ventral, dorsal, lateral surfaces. Lateral means side. Ventral, dorsal, anterior, posterior. But some lower group of animals are not having dorsal, ventral, lateral surfaces. Their body is either cylindrical or elongated. I think you got it. Just imagine a steel water bottle. We have a cap opening and a lower closed part, but no dorsal, ventral, lateral surfaces. Got it? And we can cut. Just imagine if I want to cut this bottle into two equal halves. I want to cut this bottle into two equal halves. How it is possible? Just I'm drawing this bottle. This is steel bottle. I want to cut this bottle into two equal halves. How it is possible? From which plane? What is, I told you, plane is a surface. Yeah, this is one basket. See, this is round in shape, isn't it? And I want to cut this dustbin into two equal halves. How it is possible? The surface or the platform or the surface that runs through an axis is called plane. I think you got it. Plane is a flat area or the surface that runs through an axis. What is that axis? Principal axis. Now, if I want to cut this into two equal halves, from where I can start cutting? From any plane passing through central axis. I think you got it. So here, I'm just cutting this animal in cross section. See, here in this animal, the body parts are arranged in a radiating fashion. Body parts are equally arranged around the radius. So this is cross section. We can cut the animal from any plane passing through the center, give rise to two equal halves. Look at the textbook. A radial symmetrical animals can be cut into two equal halves from any plane passing through central axis. Example, Cnidaria ctenophora. Whereas in case of sponges, body parts are not arranged in a proper way, so we cannot cut the animal from any plane into two equal halves. Hence, they are called asymmetrical. They are called asymmetrical. In higher
Excuse me,
animals,
sir.
right from
Sir, so in Cnidaria and Ctenophora, they show radial symmetry, sir.
yes, they are diploblastic as well as they exhibit radial symmetry. Their body shape is either cylindrical or umbrella shape. Right. And in case of Platytochordata, the body parts are equally arranged on either side of principal axis. So, listen, we can cut the animal through how many planes, only one plane. If you cut the animal through one plane, we'll get two equal lateral. Lateral means side halves. So the name given as bilateral symmetry. Okay, na? Bilateral symmetry. So I'll repeat once again. Asymmetry. Body parts are not arranged in a proper fashion, so we cannot cut the animal into two equal halves. Example, porifera. And Nidaria, ctenophora, body parts are radially arranged, so we can cut the animal from any plane passing through central axis will give rise to two equal halves. Bilaterally symmetrical animals, the body parts are arranged in pairs. So we can cut the animal only through how many planes? One plane that give rise to two equal halves. And very, very, very important, the radially symmetrical animals are either sessile or sluggish. What is meant by sluggish? Slow moving. And free moving or swimming animals exhibit bilateral symmetry. In these bilaterians, we can find cephalization. What is meant by cephalization? Development of head at the anterior part. So they can respond to the environmental changes very quickly.
Excuse me, sir. Sir, can you explain the meaning of sessile?
Amma?
Can you explain the meaning of sessile?
Yeah. Sessile means stationary. Stationary means they cannot move. Sluggish means slow moving animals. Okay, na? Now, Charisha, you tell me.
Yes,
Dash group of animals are asymmetrical.
So can you repeat it once again, sir?
Dash group of animals are asymmetrical. Porifera. In which symmetry the body can be cut into two equal halves from any plane passing through central axis.
Bilateral.
Any plane or several planes. Radial symmetry.
Radial.
Only through one plane. Bilateral symmetry. Slow moving or sluggish or sessile animals usually exhibit dash type of symmetry.
Bilateral.
Radial symmetry. And free swimming or fast moving animals exhibit dash type of symmetry.
Bilateral symmetry.
Bilateral. In which group of animals we can find cephalization? Bilaterally symmetrical animals. Okay, ma?
Okay, sir.
All right. Next, coming to segmentation or metamerism. Segmentation or metamerism. Now, in some animals, body is externally and internally divided into partitions, what we call them as segments, with a repetition of body organs. This partition with the repetition of body parts or body organs is called segmentation or metamerism. Actually, I'll tell you one thing. Division of body into partitions is called segmentation. And these segments will have the repetition of organs serially. This type of serial repetition of body parts is called metamerism. And such type of segmentation is called metameric segmentation, found in Annelida, Arthropoda, and Chordata. Annelida, Arthropoda, Chordata. Annelida, Arthropoda, Chordata. Okay, na?
Sir, can you please repeat, sir?
I'll repeat once again. In some animals, body is divided externally and internally into different units called segments, with a repetition of body organs. Such type of a repetition of organs is called metamerism, or the segmentation is called metameric segmentation. Metameric segmentation. Okay, na?
Sir, so what's the difference between segmentation and metameric segmentation, sir?
Yes, I'll tell you one. Division of body into different partitions by division of body into different chambers by means of some partitions are called segments. Now, these segments are repeatedly arranged with the repetition of some body organs. Did you completed structural organization in your school? Structural organization in animals. Not completed? Okay. Anyway, I'll explain it later. For example, nephridia, excretive structures are repeatedly arranged. Ganglia are repeatedly arranged. So, repetition of body parts along with the repetition of segments is called metameric segmentation. Metameric segmentation, found in Annelida, Arthropoda, Chordata.
Sir, can you just repeat the definition you gave, sir, for metameric segmentation?
Yes. In some animals, body is divided into segments with the repetition of body organs. A repetition means the same structure will be repeated several times. And even in humans also, we can find metameric segmentation, but it is restricted to internal body parts. Can you guess some structures which are found in our body? See, our body is not divided into partitions externally, but internally we have repetition of body parts. You can guess those structures very easily. Anyone? Ritwik?
So,
Can you guess some of the structures who are repeatedly arranged? Even human beings belong to the phylum Chordata, and we're also having segmentation, but that segmentation is not clear externally.
sir, is it
Or even internally, there is no partition, but we can find metamerism.
sir, is it spinal spinal cord?
Very good, very good. In spinal cord, we can observe vertebrae. Vertebrae are repeatedly arranged. That represents segmentation. Number two, ribs are segmentally arranged, but that segmentation is not clear. But still, we can find some repetition of body part. That is called metamerism. I think you got the difference between metamerism, metameric segmentation. Amma, division of body into partitions is called segmentation. If these segments are repeatedly arranged with repetition of body parts is called metameric segmentation. I think you got the difference. Is it clear, Amma? Charisha? Levin?
Yes, sir.
Yes, sir.
Right. Okay, very
Yes,
good.
sir.
Very good, very good. Next, coming to notochord. Just I'll stop with this notochord and we'll continue from tomorrow onwards. I'll start class from 7. Are you comfortable at which time? You tell me. 7 or 7:30?
So
Which is
7:30
best?
is better.
7:30 or 7?
7:30.
7:30 to 9.
Yeah, sir.
Okay, Amma? Is it okay for all?
Yes, sir.
Right. Okay.
Yes, sir.
Right. Okay. Next, notochord. This is elementary canal. And in some animals, we can find this is pharynx, gut, anus. This is mouth. And above the gut means elementary canal. We can find a stiff elastic rod that gives support to the animal. This is called notochord. In some animals, the notochord is replaced by vertebral column. Above the notochord, we can find a nerve cord. And the pharynx is having slits, means partitions or small slits called pharyngeal gill slits, where we can find gills. I'll explain all these things later. Those who are having notochord are called chordates. Without notochord are called non-chordates. At the beginning of this chapter, we discussed about the non-chordates and chordates. Who are non-chordates from porifera to hemichordata. And rest of the animals come under chordata.
Excuse me, sir. Can you please explain pharyngeal gill slits, sir?
Pharyngeal gill slits are the slits. Slits means small cleavage. Slits confined to pharynx. They are internally lined by gills. So what happened here in chordates? The water moves into the pharynx through mouth and leaves through gill slits. See, this is small basket. This is small basket is having slits. Just imagine this is pharynx having slits lined by gills. The water move into the pharynx through mouth and leaves through slits. While the water is coming out, the exchange of gases takes place at gills. And very, very important, notochord is derived from mesoderm. Nerve cord is derived from ectoderm. Pharyngeal gill slits are derived from ectoendoderm. So these are the three basic fundamental chordate characters: notochord, nerve cord, gill slits, and also we can observe post-anal tail. That means a tail present behind the anus is called post. Post means after. Post-anal tail.
Sir, can
Okay,
you
ma?
please repeat one time, sir?
Okay. Notochord is a stiff elastic rod located above the alimentary canal that gives support to the animal. Number 2. Nerve cord is present above the notochord. In higher animals, it is differentiated into brain and spinal cord. Derived from ectoderm. Gill slits are derived from endoderm and ectoderm. Okay, how they are formed, I'll tell you. Just watch the animal from surface view, means dorsal view. See, this is animal. This is pharynx. Okay. Body wall, alimentary canal. I think you know body wall derived from ectoderm, alimentary canal, inner lining, endoderm. See, during development, small pouch-like structure develop from pharynx. Likewise, this body wall invaginate means in pushing. Both meet together and leave a gap. This is called pharyngeal slit. A pharyngeal slit is formed by the joining of endoderm and ectoderm. Okay, ma? This is about the introduction in lesson. And from tomorrow onwards, we'll start phylums, Porifera, Nidaria, everything, and how to remember these phyla. Everything, I'll just tell you the techniques, how to recollect, how to remember. And one thing, the more you practice, the more perfection you'll get. And imagine this is not a conceptual part. This is simply a memory part. So I think in your childhood, you might aware of Twinkle, Twinkle, Little Star. I think everyone might sing that particular song, Twinkle, Twinkle. Actually, in your childhood, we'll sing Twinkle, Twinkle, Little Star with action. But actually, we don't know the meaning of a single word, isn't it? But still, we can sing with action. How it is possible? by continuous practice. And here also, if you really want good marks or the best marks in biology, especially zoology part, definitely I'll teach in a proper way. I'll tell you some tips and techniques to recollect, to recognize, or what we call to recollect the subject. The more perfection you'll get, the more success you can achieve. So definitely in coming lessons, in coming classes, definitely I'll teach you how to get success. And if you follow my teaching, if you follow my words, definitely I can give guarantee of 180 out of 180 in final NEET examination. That guarantee I can give. And you people are very good, and you're listening with proper concentration. And of course, the memory and concentration is also very nice. And I appreciate your patience. And can I ask a few questions before closing this class? Who is ready to give the answers? Yes. Number one. Amma? Who is ready? Krutvik, Levin, Cherisha, or Evelyn? Right. I'll ask few questions. First, triploblastic animals. Platyhelminthes. I'll do one thing. You just go through the textbook and you come with full preparation. And immediately after the beginning of class, I'll ask questions. Not randomly, individually. Every day I'll ask questions. Okay, Amma? Are you happy with my teaching?
Yes, sir.
Yes.
Not happy? But just remember one thing. Already you listen this concept, isn't it? Because of that reason, I'm just moving a little bit fast. Okay, but later on, definitely I'll go in a particular pattern and try to make you perfect in zoology part. Okay, Amma? So thank you and have a nice day. Good night.
Thank you, sir.
Thank you, sir. Bye, sir.
Thank you,
Okay,
sir.
Amma.
Thank
Good
you,
night. We'll meet again tomorrow at 7:30 p.m. Right, Amma?
Yes, sir.
Okay, okay, bye.
Thank you very much, sir.
Thank you, sir. Thank you. Your students are very nice.
Thank you, sir. It's now our students.
If the students are like this, we can enjoy teaching.
Sure, sir. Sure, sir. I'm sure they're all focused in getting NEET under your guidance.
Definitely,
Zoology
sir.
part is taken care of.
Definitely. If the students show that type of interest, definitely I'll make them perfect in my zoology part. It's my promise.
Thank you, sir. Thank you so much.
And along with NEET practice, I'll give assignments.
Definitely.
For every chapter, every part, I'll give assignments and practice papers, everything. And we can conduct examination papers, everything.
Sure, sir.
Okay, sir?
Thank you so much, sir.
Okay, sir. Thank you. Thank you very much. Amma, good night, all of you.
Good night, sir.
Thank you, sir.
Thank
Good
you,
night,
sir.
sir. Bye,
Thank you,
sir.
sir.
Thank you, sir.
Okay,
Good
now. Okay.
Yes, sir. Thank you, sir. Excellent.